So I'd like to start by thanking Burnbrae for sponsoring this next set of sessions. Thank you very much to Burnbrae. To get started, I'd like to introduce our first session now in our behavior and welfare section. Our first talk today is Domesticated. How does artificial selection influence the brain of domestic fowl? Audrey Guyonnet, University of Lethbridge. Go ahead, Audrey.
Thanks, Vicki. Let me just share my screen. Okay, can everyone see that?
Yes.
Looks good.
Okay, perfect. All right, so thank you so much for attending my talk today. My name is Audrey Guyonnet, and I'm a second-year master's student at the University of Lethbridge in Alberta, Canada. My work is on domestication and how it has altered the brain of domestic birds. So animal domestication is widely agreed to be the milestone that marks the beginning of modern human civilization. For thousands of years, we've artificially selected a number of desired traits across species, usually with the goal of improving commodity yield, like meat, egg, or material production. From a behavior standpoint, domestic species are universally selected for tameness. So tame animals are less fearful of humans, making them easy to handle and house in large groups. But we've also selected for a wide variety of behaviors among performance or working animals, enhancing them far beyond the natural behaviors of their wild ancestors. Comparisons among domestic breeds can yield valuable information about how artificial selection can cause lasting changes on the behavior and morphology of these species. So for instance, in about 8,000 years of rock pigeon domestication, we've created over 350 unique breeds with more trait variation than any other avian species studied to date. Show breeds demonstrate the bulk of the morphological variation. For example, fantail pigeons can have up to 40 tail feathers as opposed to the usual 12 to 14. However, pigeons aren't only bred for morphology, and numerous sporting breeds are also selected for a diverse number of in-flight behaviors. The homing pigeon is perhaps the most famous example of artificial selection for a behavioral trait. Homing is a behavior originally used by wild rock pigeons to return to their cliffside nests over visually stark landscapes, and involves integrating multisensory cues from the environment to map the most direct route home. Today, the domestic homing pigeon can locate a home loft from 1,000 kilometers away, making them incredible spatial navigators. But after thousands of years of selection for homing, what kind of trends can we expect to see in the homing pigeon brain? So relationships between brain and behavior are often explained by the adaptive specialization hypothesis, which states that changes to cognition are measurable by associated changes in neuroanatomy, like regional brain volume or neuron number and density. In domestic species, selection for tameness and subsequent loss in behavioral complexity have been followed by corresponding reductions in brain size. Not all behaviors have been lost. Many, like navigation in pigeons, have been enhanced by artificial selection, meaning we might see the opposite trend to the brain regions that control those behaviors. For this question, I've focused on the homing behavior in pigeons. Like other navigational processes, homing is controlled predominantly by the hippocampal formation. So I hypothesized that selection for homing might place greater demands on this brain region and predicted homing pigeons to have larger hippocampal regions with more neurons than non-homing breeds. I conducted an interbreed comparison of show, sporting, and feral pigeons, and feral pigeon populations are not true examples of a wild type, but share life history traits with ancestral wild rock pigeons. So for volumetric analyses, I quantified regional brain volumes using stereo investigator software. I looked at the hippocampal formation, which is made up of hippocampus proper and area parahippocampalis, and is largely involved in spatial cognition. I found a significant difference in HF volume across breeds, even after accounting for differences in telencephalon volume. Homing pigeons average a 9 to 21% increase in HF size, but I found only a single pairwise difference between homers and high flyers. The only exception to this was the Norwich Cropper, a show breed we'll continue to discuss due to its consistent role as an outlier. So the next measures I analyzed were neuron number and density. For these, I fluorescently labeled functionally mature neurons using a new antibody and counted them in stereo investigator. So I found significant differences in both absolute and relative HF neuron numbers and density between breeds. Specifically, homing pigeons have experienced a 41 to 56% increase in average HF neuron number and a 38 to 50% increase in density in comparison to feral pigeons, active breeds, and capuchin show pigeons. So here's a visual of the HF in homing and feral pigeons. As you can see, there's little difference in volume, but a huge difference in neuron density. On the left, the homing pigeon demonstrates incredibly high neuronal density, and in comparison, the HF region of feral pigeons is barely visible, while the archipelagium below it is completely lit up with neurons. These results suggest that neuron populations are likely more plastic than volume. So when improving cognitive power, it may be less energetically costly to increase neuron number as opposed to the size of an entire brain region. Despite being incredible navigators, homing pigeons don't display dramatic neuroanatomical differences from all other domestic pigeon breeds, like I expected. And I think it's possible that breed introgression plays a large role in this finding. So many of the show breeds we used, like show rollers and show homers, are recently derived from their sport-bred counterparts. And while these animals are now selected for conformation instead of behavior, they're frequently crossed back to homing pigeons for desired traits. In comparison, sporting pigeons like roller or high flyer breeds are kept genetically distinct to preserve unique behaviors. But I was also interested in knowing if increased neuron expression was unique to the HF region in homing pigeons, or if it was a non-specific change influencing the whole brain. So as a comparative structure, I chose to examine the avian septum due to its importance to limbic system functioning. The limbic system controls emotional response behaviors, such as those related to aggressive and sexual displays. In domestic species, selection for tameness and other behaviors have drastically altered emotional responsiveness. This project was primarily exploratory, and I expected septum might differ between pigeon breeds selected for different behaviors, and that these changes might differ from trends in HF neuroanatomy in the previous chapter. So for this comparison, I once again used a variety of show, sporting, and feral pigeons. Notably, I included Norwich croppers, shown here on the left, a breed selected for exaggerated sexual characters like an enlarged crop, upright body posture, and elaborate sexual displays. Because the limbic system controls sexual behaviors, I expected intensive selection might positively influence cropper septum neuroanatomy. So croppers display significantly larger absolute septum volumes than all active breeds and feral pigeons. Interestingly, and contrary to trends in the HF, I found homing pigeons to have smaller relative septum volumes than all show breeds, as shown in the scatter plot on the right. So results for septum neuron number and density were a bit more variable. The top left graph shows absolute neuron counts, and significance from croppers is demonstrated by the orange asterisks, whereas significance from ferals is in blue. So cropper pigeons had significantly higher absolute neuron counts than most breeds, whereas feral pigeons were significantly lower. And that latter trend was followed in the scatter plot to the right, where feral pigeons display significantly lower relative neuron counts as well. And then finally, feral pigeons also had lower neuron densities than some, but not all of the domestic breeds. So here's a quick summary for our breeds of interest. Homing pigeons display increased neuron number and density in the HF region. These relationships are independent of changes to other brain regions, like the septum, that decreased in volume and showed no significant difference in neuron population. Despite high genetic similarity to homing and other domestic pigeons, feral pigeons demonstrate decreased neuron number and density across all regions of interest. And finally, Norwich croppers show increased neuron number across all regions, in addition to increases in septum and total telencephalon volume. So as expected, homing pigeons selected for HF-dependent spatial cognition traits display region-specific increases in HF density. Quite possibly, selection for homing has positively influenced genes that upregulate neuron proliferation and survivorship in the HF. In feral pigeons, an overall decrease in neuron number may suggest that chronic stress of free living, particularly in urban environments, can negatively influence whole brain neurogenesis. And finally, cropper pigeons selected for exaggerated sexual displays demonstrate an overall increase in neuron number and density across brain regions. HF and septum are part of the limbic system, and selection for sexual characters may have increased neuron expression across both regions. Interestingly, we found no overall trends by broad groupings, such as by comparing feral versus show versus sporting breeds. Instead, specific breeds display either region-specific or brain-wide differences in neuroanatomy, suggesting that artificial selection for specific behaviors can differentially alter brain regions. Future work in this area could look at the importance of practicing behaviors during development to the survival of neural networks in the avian brain. So finally, I'd like to thank NSERC, CFI, and the University of Lethbridge for making this research possible. I'd also like to thank all the members of my lab for their help with this work. Thank you all for listening, and I'm happy to take any questions you might have.
Thank you, Audrey. That was fascinating. Just a reminder to all of you that if you have any questions, please place them in the question and answer box. And seeing how this is behavior and welfare, I encourage everyone to improve your posture or stand up if you're not on camera. I will not do that, but I will put my shoulders back and adjust my back. So again, if you have any questions, please enter them. Audrey, I'm just wondering, who can use this information? So you showed that the neuron density was increased, but the volume stayed the same. Who would be interested in that information?
Yeah, so that's a very good question. So this kind of research is mostly looked at in wild animals, but domestic species are an incredible model for looking at how natural selection would influence species and change their brain over time. It's just that we are an incredible version of natural selection that works so fast. So we can look at how the brain changes over time, but at an incredibly rapid rate. Like 8,000 years is really nothing when you think of selection. But I mean, we've created more variation in dog breeds, let's say, in the past, I think, 50,000 or 5,000 years than the past 30 million years of natural selection. So it's really a good way to look at how behavior and anatomy can change over time. And then also by looking at neurogenesis and neuron survivorship, you can get a feel for, as how I looked at stress responses in those feral pigeons. If your animals are stressed, you're going to have lower survivorship in the brain. So you're going to know that something is influencing them negatively. So there are a few different aspects of how you can look at it.
Excellent. I'm glad I asked. So we have two questions. Were you surprised by the difference between feral and homing pigeons? Having been a pigeon keeper in my youth, I know that there was a loss of homing pigeons to wild flocks.
Yeah, that is why that result is so exciting, because the two groups that are the most genetically similar would actually be homing and feral pigeons. Feral pigeons are relatively new to North America, especially. They've been here only for as long as we have, and feral pigeon populations are made up entirely of escaped domestic pigeons here. So I think that the genetic similarity is something like, I can't remember the exact number, but they're incredibly similar. So that then to find such a drastic difference between the two was absolutely astounding. Very exciting.
That's awesome. And I can tell you're excited, and that makes me happy. That's good. Question, was it difficult to source the information from so many different breeds?
Absolutely, yes. I have read so many books from the 1800s to early 1900s, back when pigeon keeping was very, very big. Specifically, Darwin was most interested in pigeons. And when he wrote his book, The Variation of Animals and Plants Under the Influence of Domestication, he mostly focused on pigeons, because we originally had thought that they were so variable between breeds that they had to be from multiple different lineages, like they had to have multiple different ancestors. And he was the first to realize that actually they only had one common ancestor. And if you breed any, despite how different they look, I mean, if you saw the picture that I have where I show the variation between them, if you interbreed any of them together for long enough, you end up with something that looks like a feral pigeon, with the classic blue bar look and the gray feathering. So it's definitely hard to find the information, because a lot of it is word of mouth. So we also did a lot of kind of interviewing local pigeon breeders to see what kind of behaviors they've seen in their birds and what is common, because a lot of it is, it's generational and people just passing the information between themselves.
Thank you, Audrey. We are ready to move on. We will leave it there.
Yeah.